The overall goal of the following experiment is to establish an in vitro assay for examining the role of hormones, cytokines, and/or growth factors in driving cancer cell invasion. This is achieved by determining whether there is a difference between the invasion index of different cell variants or mutants. As a second step, the invasion assay is repeated using charcoal-stripped media, which removes any hormones, growth factors, or cytokines from the serum. The invasion assay is then repeated with the addition of a specific known factor to the charcoal-stripped medium to determine whether migration toward that factor enhances or inhibits invasion. The results show the changes in the invasion index for specific cell populations that occur during migration toward known experimental factors based on the normalization of the results against the invasive index of non-invasive cell populations.
I first got the idea for this method when I was performing a traditional invasion assay, and I noticed that there was a significant difference between the invasion indexes for different cell mutants.
The main advantage of this technique over existing methods, like the traditional invasion assay, is that with this technique, you can specifically investigate the role of hormones, specific growth factors, and cytokines in promoting or inhibiting cell invasion.
This method may be helpful in answering key questions in the field of cancer research, including which factors lead to cancer metastasis.
The implications of this technique may provide a therapeutic approach to future drug design.